Chapter 22, Part 2, Screencast for the Immune System. This is the final screencast for the immune system, and the topic is going to be immunologic memory. Now, memory results from the formation of a long-lived army of lymphocytes upon immune activation. Now, adaptive immunity activation requires contact between the lymphocyte and the antigen. There is a lag time between first exposure and direct contact. The activation actually leads to the formation of many memory cells against specific antigens. Here we can actually see the difference. The blue is actually the lag phase, and on the y-axis we're actually looking at serum antibody titer. We're going to be primarily concerned with IgG. So notice, from the first exposure to the antigen, there is a pretty significant lag time of 3 days or so approximately, but also we can look at the antibody response, specifically IgG. And remember, this is the more general of the immunoglobulins and actually has more functions, and it's the one that lasts for approximately 23 days once produced. Now this has been the first exposure, and now here we have the secondary response. So this will be re-exposure to the same antigen, and notice the big difference. There's hardly any lag time, and the exponential difference in how much IgG is actually being produced during this particular exposure. So what we can see here is memory really is important, because that first exposure is what actually is going to protect us the next time we encounter that particular antigen. We can see by the exaggerated response on the second time around. Now, here we're going to talk about active and passive immunity. And when we talk about active immunity, that is the production of memory cells due to contact with antigen. So there is an antigen challenge in this situation. You have naturally acquired, and that is direct exposure to antigen following entry of the pathogen into the body naturally. So I contracted chickenpox as a child. I was not vaccinated for it, so that would be an example of naturally acquired. Artificially acquired is exposure of the antigen through a vaccine. So those of you that have been vaccinated for chickenpox, that's an example of artificially acquired. Now, passive immunity, now there's no production of memory cells. There is no engagement with the antigen, and the antibodies from another person or animal are being passed on. If we look at naturally acquired, transfer is from the mother to the child across the placenta or in breast milk. Artificially acquired, this is the transfer of serum containing antibodies from another person or an animal. An example might be antitoxin or antivenom for a poisonous reptile. Now, when we look at what vaccinations actually are, they are weakened or dead microorganisms or components, and they're designed to stimulate the immune system to develop memory B cells. They provide a relatively safe means for initial exposure to microorganisms. If later exposed, this is when the secondary response is triggered, and the immune system responds predominantly from the humoral branch. Now, generally, they may provide lifelong immunity, or there are some vaccines which will actually require a booster. Now, HIV and AIDS is acquired immunodeficiency syndrome, and it is a result of the HIV virus. And what this virus targets and infects are helper T cells. It resides in body fluids of infected individuals, and generally it can be transmitted by intercourse, needle sharing, and breastfeeding. Diagnosis of AIDS is when helper T cells count drops below 200 cells per cubic milliliter. Death usually occurs from opportunistic infections or cancer. It can be prevented through safe sex. Currently, there is no vaccine against HIV, and what they do now is actually treat it in such a way that the viral load is so low that it cannot be passed on through any type of sexual activity.